Hello and welcome to the Light-Sheet Chronicles. I'm Dr. Elisabeth Kugler, a science communicator and your host of today's episode, Image Analysis in Light-Sheet Microscopy, Big Data Processing, Handling and Storage. In this episode, we will talk about biomedical image analysis in light-sheet fluorescence microscopy, including the handling, processing and storage of big data. And we talk about this because light-sheet microscopy opens the door to stunning 3D views of biology, but it also comes with a major challenge, which is data. Datasets can quickly grow to terabytes in size, making them very difficult to handle, process, and store. And beyond the sheer scale and size, scientists also face hurdles in ensuring data quality, reproducibility, and accessibility across different teams or locations. And so in this episode, we'll explore how to tackle these challenges in biomedical image analysis and what it really takes to make sense of light sheet data. With us today, we have two amazing guests. First, we have Dr. Kate McDole. She's a research group leader at the MSc Laboratory of Molecular Biology, or MSc LMB in Cambridge, and her lab explores the morphogenesis of early mouse embryos using a combination of advanced light sheet microscopy, biology, computational methods, and biophysics. So welcome, Kate.
Nice to be here.
It's fabulous to have you on board. We really look forward to this conversation. And with us today is also Dr. Jan Roden. And Jan is a senior software architect at Bruker, the Light-Sheet division of Bruker. After studying physics in Germany, he has moved to the US for a postdoc in theoretical physics at UC Berkeley and Berkeley Lab. And since 2016, he is software architect at Bruker. So welcome, Jan.
Thank you. Thanks for having me.
It's fabulous to have you both. And so we start off really with a very general topic around biomedical image analysis and data structure. And so, Jan, maybe you can start us off by giving us a very general introduction to what some particular challenges are in light sheet microscopy with respect to image analysis and data.
Yeah, I'd love to. So, light sheet microscopy is a quite versatile imaging method, right? Because it's so fast and also very gentle on the sample in terms of light exposure. And so therefore it has a very broad range of different applications in biology and medical research. And so, for example, people use it to image organoids, or it's used a lot in developmental biology or cancer research, and for a very broad range of different sample sizes from whole mouse embryos, plants, zebrafish, Drosophila embryos, but also just looking at cell layers. So it's very diverse, and people use all kinds of fluorescence markers in membranes or nuclei to image different parts of the organisms. And it's used for live samples, but also cleared samples, right? And so therefore the data is also very diverse. So there's the data is multidimensional. So it has the different color channels for the different fluorophores that are imaged, but also different views looking from different sides through different objectives, and also with rotation stages sometimes from different view angles. And often especially the large cleared samples, they don't fit into a single field of view of the microscope. So the stage needs to move, and then different tiles are being imaged and in the end stitched together. And then for the live samples, looking at the development of the samples, people acquire time lapses. So all in all, it's very diverse data, very large data, right? Also because these image stacks that are acquired, they often have a few hundreds to even a few thousands of frames, which each have something like 4 megapixels, right? So as you said, Ellie, we easily have terabytes of data in size per experiment. And so processing this amount of data, of course, takes quite a long time, and the data storage can also become really a problem, network transfer, and also evaluating the data quality and managing the data. So deciding what to keep and what to delete can also become quite a challenge, especially for large facilities.
Thank you so much for that wonderful introduction about the complexity of data, because yes, absolutely. Having sat myself a lot on light sheet microscopes, I can concur. It's a lot of data, complex data. And I think we all know it's moved really far beyond just simple 2D slices, but it's 3D channels, tiles, time points, viewpoints, etc. As you mentioned, there's lots and lots of going on, like different layers to think about in the image analysis. And so I wonder, can you maybe talk with us about what's your approach to really start with analysis of multidimensional data?
Yeah. So, I mean, the most important thing, of course, is first viewing the data, right? After you acquired a nice experiment, the first thing you want to do is view the data in an ergonomic and efficient way. So it's very crucial to have a user-friendly, efficient 2D and 3D image viewer that can actually handle this data very well, and both for raw data as well as for the processed data after the post-processing. And so ideally, the data should already have a resolution pyramid, so different resolutions from the highest resolution to lower resolutions for efficient zooming of the data and 3D viewing. And also ideally, one normally would like to look at maximum intensity projections to get a good preview and overview, and for example assessing time-lapse experiments, and that's very important. So all this metadata and resolution layers, this is quite crucial. Also, if we look at the process data from, as I said, we have multiple color channels, and normally people want to overlay these and then compare them. So it's also very crucial to have good, accurate image registration, so alignment, in order to for these overlays to fit together well. And then it's important in the viewer that one can very nicely compare different views and channels, and also play time lapses. So this is, I would say, a very important instrument for first assessment of the data. And then further on for the processing and image analysis, it's, I think, very important to have software that is open and has open interfaces where one can hook in other third-party software easily, right? So, for example, the Microscope software or the post-processing software that's provided by the Microscope manufacturer, that this is very open, that one can hook in custom Python scripts and Fiji scripts or MATLAB, and also that different AI tools for cell segmentation, cell tracking, cell counting, and so on, right? That all these interfaces should be as open as possible. I think that's quite crucial, because as I said, there's a huge diversity of use cases and applications, and normally there's always some custom elements in the workflow, because each lab has, of course, their own research and experiments and what they are interested in. So therefore, it needs to be all ideally very open and modular, so that things can be plugged together and chained up as a pipeline of compute tasks, and also ideally being then interleaved again with commercial software. And so ideally, such pipelines then could also even be run already during acquisition, so that one doesn't have to wait until everything is acquired, but the processing can already start earlier. And maybe even already different processing settings can run in parallel, right? Because what ideally in the end we want to optimize is the researcher's time and get the results as fast as possible, so that overall the amount of things that researchers can look at is maximized, so to say. And I mean, open image formats, open image metadata is very well documented. Image metadata is very important, I think, so that everyone understands what all the parameters mean, and also that the processing software and viewing software, as we said, right, the data sizes are huge, so this all needs to be quite performant, and because this in the end can make the difference if you are waiting for 30 minutes versus a day for your results. So yeah, I think those are the most important points from my side.
I love that. Thank you so much, Jan, for that overview. We're only a few minutes into the podcast and we already mentioned resolution pyramids, registration, AI, cell tracking, and many more things. So that's great. Thank you. And I hope we'll get lots of time later on to talk more about these things. But that was all kind of very theoretical. And now I want to switch over to practice because we have with us Kate, who really does all of this in her lab on a daily basis. And so, Kate, I know you mostly work with long-term in vivo time lapses to study mouse embryo development, and you do that at single cell resolution over the course of days. So your data are obviously very complex and also very challenging to work through. So could you maybe introduce us to your data and your data challenges that you work with?
Yes, we have a light-sheet microscope that I built up in the lab that allows us to do a lot of custom configurations and custom imaging. And while this is nice because you can really adapt your imaging to the sample, so we can do everything from Drosophila, zebrafish, mouse embryos is what our lab works on. We also collaborate with people around the world to look at organoids, gastroloids. And so the type of data that we get off the microscope is constantly changing. And so our data pipelines that we have for both acquiring it and processing it need to be very robust and able to put in something from a Drosophila that needs functional imaging to an organoid that's been on there for 2 weeks, and be able to do so, as Jan pointed out. You want to be able to do this as efficiently as possible because some of these data sets will be 80 to 100 terabytes, and that obviously takes quite a long time to chew through. So one of the biggest features that we found that was most helpful on the microscope was we implement this real-time max projection. At each time point, the microscope will build up a max projection as it's acquiring. And that lets us have a very quick overview of how the sample's doing. Is it developing correctly? And if there's anything wrong with the experiment, then we just end it right there. So rather than continuing to acquire data that we'll have to process, if we know something is going wrong, we can stop it ahead of time and put on a different sample. Or if we can look and see there's a certain time point we need to be acquiring, or if we need to feed it, we know where we are in terms of its development. So that allows us kind of some real-time processing just to see how the sample's developing. And then once we get the data off of the host computer, so we need to write these things onto the computer fairly quickly. So we kind of use intermediate computers to move things off of the cameras onto the microscope. And then that puts it onto the network so it doesn't interfere with the operation of the acquisition machine. The initial processing was to try and clean up as much, I don't want to say unnecessary data, because I don't think there's any such thing as unnecessary. Well, there is. Such things as like removing background. If you're looking at a mouse embryo at the early stage, it's quite small, but you're still using the full camera chip. So you can zero out a lot of that background, and that actually reduces the size of the files quite considerably, and it also speeds up on the processing time as well. So yeah, if you're dealing with things like really, really large, say, cardioids, which are hollow on the inside, sometimes fusing it isn't necessary or is actually quite complicated. But if somebody just wants to look at one side of the cardioid because that's or the organoid where something is happening, then you don't necessarily need to fuse it, and you can reduce the size of the stack just by cropping it. And so you can do your other kind of hot dead pixel corrections just on that, and that's very simple. Our biggest problem is the visualization of these data sets. Is you want to look at a full time-lapse data set of something that's been, developing for 2 weeks. This ends up in a very, very, very large file. So looking at the max projections over time, but then if you want to start to look at the 3D processes, there's not a lot of software out there that handles it very well. So you ultimately just have to be patient with a lot of these data sets, and realize anything you're going to do for them, from opening them to visualizing them or making a movie, will take days and in some cases weeks just to make a movie.
Absolutely. And I think that's also one of the biggest challenges, this kind of time consumption. Like you say, even just the rendering just takes absolute ages. And I love what you mentioned about like empty data and like hot dead pixels, because I think, like you say, there's so much empty data that we still work with. I know when I did my PhD, I worked in the brain vessels of zebrafish. And so I think about only 8% of the data that we acquired were actual data of the blood vessels and the rest was just empty. And so, as you say, there's a lot of kind of empty data that you want to remove. But the other thing that you mentioned is just real-time processing. And I think that's getting more and more important, especially also to free up the microscopes so people can run longer experiments and kind of really make the most use out of the microscopes that they have. And so I know that you mentioned this real-time processing, but also I know that you're very interested in event-driven microscopy, Kate. So maybe you can tell our listeners why this is really important and what you're working on.
Right. So event-driven microscopy was kind of coined by Suliana Manley. So they put out this paper in 2022. It was a nice demonstration of event-driven acquisition, where the idea is basically you let the microscope make decisions as to what's happening to the sample. So you can give it a user-defined event, such as in the case they were doing mitochondrial and bacterial divisions. You could look at cell divisions. You could look at the appearance of a fluorescent reporter. So maybe you're looking at a long-term development of a sample. And you want to track these cells, but at a certain time point, these specific cells you're interested in turn on a reporter, and then you want to image much, much faster, or maybe you want to zoom in and get at much higher resolution. And most of the time that anything interesting or bad happens to the microscope is between midnight and 4 a.m. I have actually calculated this. It actually is 80% of the things that go wrong happen between midnight and 4 a.m. So you can't rely on humans to intervene and catch all these events, or they're not either not fast enough, or you just can't be babysitting the microscope for 2 weeks straight. And so event-driven microscopy is either using, kind of user-defined modules or frameworks, or using machine learning to then identify. So it takes the image off the camera, processes it, looks for the features that you've told it to look for, and then it reacts to it. So either by increasing the acquisition speed, zooming in, ablation, photo conversion, and then going back to a normal imaging mode. So depending on what it is you want it to do. And so this is quite nice if you have developing samples that are changing or samples that are behaving, and you want to capture this behavior in a different way or at a different resolution. And so this is kind of what we drive a lot of the microscope towards, is this kind of autonomous event-driven microscopy. Is that the microscope, the way I built it, is that it can run almost completely autonomously for weeks at a time. You just need to go in and check on it, check on the sample, maybe feed it if it needs feeding, depending on the sample, and all of the degrees of freedom on the microscope, that basically everything that we could put a motor or a wire on, we stuck a motor and a wire on, and that's all controlled by the software. And so it can adjust, everything from the field of view. If the sample moves, it tracks after it, it expands the Z range around it. It'll adjust the light sheet to keep the sample to keep the best focus, keep the light sheet optimized in the best position, correct for things like refractive index mismatches, all sorts of things that it can do. So the idea is to capture the best image possible, and then also be able to do other events as the embryo develops.
Oh, I so want to come to your lab and see all of that. I'm like, oh my God, it sounds amazing.
It doesn't look like a microscope. Anyone who walks in there is like, this is not a microscope.
It's a spaceship. Absolutely. And I also love what you said, that 80% of things go wrong at night. And I think, everyone who's run a time lapse can concur. It does. Yes.
But I have actually calculated it, and it does. Statistically, it does not make sense. But 80% of the things that go wrong are between midnight and 4 a.m.
Oh, I love that. We need to see those numbers. That's great. Absolutely. But I also love that you kind of started, talking about this on-the-fly switching and event-driven microscopy. And a lot of it is probably human-based decisions in general, and boundary conditions that we set in terms of like hard coding, probably like tracking, etc. But I think also with AI, this probably changes a lot, right? And a lot of these kind of decisions can be automated. What do you think about that? Kind of, where's the future heading, and where are we maybe at the moment?
Yeah, and so that was some of the stuff that Suliana did as well, is to use these neural network-based recognition frameworks. And so the idea is that can you train a network to recognize a certain feature or a certain event, and then it can pick it out of the image as the microscope's acquiring it, and then respond accordingly. So it knows if you're looking for, I want this specific cell type when it divides, I want you to recognize, before it starts to divide, and then be ready to, zap it or image it at a faster speed or do something else, take a recording off of it. So you just let the microscope, do its thing, and it will acquire the images, process them, and then the network should be able to identify these events and respond to them. And so it adds a lot more complexity on the acquisition side. But especially when you're working with animals, being able to capture the most from these image data sets reduces the amount of embryos and samples you have to put on and the number of animals that you ultimately use. So,
which is always great, absolutely, with the 3 Rs. So that's lovely. Thank you so much. I should have known you during my PhD. It would have saved me a lot of time. And so today we have talked about biomedical image analysis and light-sheet fluorescence microscopy, including processing, handling, and storage of big data. And so to our listeners, if you want to know more about biomedical image analysis for the Bruker Light-Sheet Microscopy division, make sure to check out our Lux Bundle Tech Note, which is linked below. And so to our listeners, many thanks for tuning in.
This podcast was sponsored by Bruker Luxendo GmbH, the Light-Sheet division of Bruker's Fluorescence Microscopy division. Bruker is a manufacturer of scientific instruments for life sciences, materials research, and industrial or applied analysis. If you'd like to learn more, please visit www.bruker.com/light-sheet. This podcast was produced by Keep It Science.